Good afternoon, and I'm Alessandro Marcello. I work at the International Center for Genetic Engineering and Biotechnology, which is an international organization with headquarters here in Trieste. I'm a virologist by training, and I've been working on several aspects of virology during my career here at ICGEB. So really, what we do in the lab are different aspects of virology, from basic research in viral replication and cell response to infection, to more applied and translational research related particularly to diagnostic and surveillance of viral infection, as well as the development of antivirals, so antiviral drugs. Then we work on, of course, on mechanism of action of the drugs and also on the detection of emerging viruses, kind of reconnecting to basic virology. So we tend to have a program where we mix aspects of basic virology and translational virology together. My group has a very high turnover of researchers from different parts of the world that bring their own expertise and share with us their experience with different viral infection in different parts of the world. Of course, before 2019, my work has developed basically around HIV for most of my career until, let's say, 2010. And then more recently into flaviviruses, which are viruses that are delivered by ticks or mosquitoes, and are also relevant for what's going on. Okay. That are also relevant here in Italy, more and more relevant for the south of Europe because of the introduction of new species of insects. Our lab is mostly dedicated to molecular virology, meaning molecular aspect of virology, particularly the use of molecular tools and cellular tools for viral infection. We also operate the so-called BSL3 facility, which is a containment facility for the manipulation of pathogenic viruses, and which is basically a containment facility where you can operate in an environment that does not allow the escape of potentially infectious material outside of the laboratory, plus a series of procedures and protection for the operator for being able to do the experiments over there. So, of course, in 2020, we had the epidemic, the SARS-CoV-2 epidemic. So we started working also on this virus from the very beginning. And until basically for 3 years, we focused mostly on SARS-CoV-2. And then in the last year, we picked up again the work on flaviviruses, together with some ongoing work on SARS-CoV-2. Okay, so today, the purpose of this talk is to give an overview of the activity of the lab. And to do this, I will focus on SARS-CoV-2 because this helps me in having a common theme during the different slides. But of course, this is also can be expanded to other viruses, because more or less we have the same kind of approach for different viruses. Okay. So what happened in 2020? We all know that we had this outbreak of SARS-CoV-2 in China. I was in China in January of that year. I was talking with my colleagues, and they were mentioning this new infection in Wuhan. So when I came back, I started monitoring the news more closely to understand what was going on. And as soon as I realized that the infection was moving outside of China, so it started to be detected also in different in other countries, I was 100% sure that it was going to hit also Italy sooner or later. So together with colleagues also in area, with Danilo for the genetics and in the hospital for the diagnostics, we started to set up protocols for the handling of SARS-CoV-2, of coronavirus. As you know, we are an international organization, so our interest is not only local at the level of the country that hosts us, like Italy in this case, but we are also interested in our member states that are worldwide. And their requests are for us very important, because we try to address problems that occur in different situations, in different countries with different capability to be able to perform diagnostic or surveillance of viral infections. So what we set up to do was to support the local, so in Italy, and global diagnostic and surveillance for this virus, to develop molecular tools for the detection, so new molecular tools or reinforce non-molecular tools for SARS-CoV-2 infection, and as well, not only molecular, but also serological, so not only to detect the virus or the genetic information of the virus, but also to detect the immune response to viral infection, support to industry for inactivation procedure, and identification of antiviral drugs, and plus understanding the mode of action, and understanding in general the cellular response to infection, which is the basic aspect of our research. Of course, in the context of an ongoing outbreak of a virus, the basic aspects were kind of secondary with respect to, at the beginning, particularly to reinforce the ability to detect and survey the virus. So the first thing that we did when we had the first cases here in Trieste was to isolate the virus. So what does it mean? This is a very basic biological technique that is very old-fashioned. It's not very fancy. You have to grow the virus in cell lines. You don't know if the virus will grow, so because this is depends on the virus. There are several viruses that don't grow in cell culture, but most of the viruses would do. So we have a cell line, which are vero cells that are monkey cells that are defective of the immune response to the infection, the innate immune response to the infection, and they're very prone to be infected by different kinds of viruses. So we took cells and we incubated specimen from the hospital, specimen that we, and together with our colleagues in the hospital, already detected by RT-PCR to be positive for SARS-CoV-2. So if you infect cells, you can see here two examples. These are pictures taken in the laboratory with my mobile, actually. And you can see vero cells that are mock infected, so you have a perfect monolayer. If you infect with swabs from an infected individual, you start seeing cells growing, rounding up and detaching from the plate. This could be anything, could be whatever contamination. Then you have to passage this in another fresh cells, and also confirmed by PCR and confirmed also by sequencing. So this is the first procedure for isolating a virus. We isolated 4 and then more and more viruses from patients. And this is very important for 2 reasons, because at the time, we didn't have a method for sequencing the virus that was the one that is used now, which is targeted sequencing, so already set up for targeted sequencing of SARS-CoV-2. We had a method to sequence everything, so it was less sensitive and more difficult to do it on samples, but better on a virus that is already grown into cells, so you have a bigger amount of the virus. And also because having a virus, so an infectious particle at hand, it's the most important thing for doing any kind of research on this virus. If you want to develop an antiviral, you need to test this drug against a virus, so a virus that is infectious, as well as if you want to study the molecular biology of the virus. All our activities were made public through a web page and a resource page that was free for our member state, but also outside of our member state. So everybody could have applied, and they actually did apply to us for reagents that we developed during this program, for protocols and anything that we have developed. It's a bit slow, okay. So as I mentioned, one of the first things that we did was to sequence the virus together with Danilo here in area. And as I said, initially we did the sequencing on whole viruses. And this was an amazing story, because basically in 2 weeks from isolation to publication was very fast track. Actually, was a record time for a paper to come out. Actually, I remember I submitted it in the evening. In the morning, I had already the reply that it was accepted in Journal of Virology. So this never happened in my career, and it will never happen again. That's meaning how people were eager to know what was circulating. I remember that Italy was the first country in Europe to be hit by the virus. So we want to know what was circulating in Italy. Of course, in other parts of Italy, other people were doing the same kind of work, but we were probably the first to do the identification of the circulating virus of the time, which was of course, SARS-CoV-2 derived from the Wuhan or ancestral strain, but already with a mutation, the D614 mutation that is characteristic of the wave of infection at the very early phases here in Europe. So already a variant, if you wish, but at the time was not even called a variant, it was just Wuhan, the SARS-CoV-2 virus. Now, this first initial process went on with in time by isolating the virus in the different waves and sequencing the virus in the different waves. Then, of course, this became a standard procedure in the health system of Italy and Europe. So now it has been picked up by the hospital, and so it became a routine procedure for analyzing the circulating variants. So we dropped out of that because we didn't want to do a routine sequencing analysis because that was taken up by others. But we wanted to focus on our role as in support of our member states. So we responded to the request of several countries to help them identify what was circulating in their countries. And this is important because, of course, you may have hub of sequencing like Europe or South Africa in Africa or US and so on that are very potent and able to sequence many different viruses circulating. But of course, this has to be also geographically distributed. So you need to know in each country what's going on because a variant can emerge everywhere. So it doesn't necessarily emerge in a place where you have high sequencing. You may emerge in another place, and then the delay from the first case of infection of a new variant to be able to be recognized in a country that has the potential to sequence is critical for the managing of the variant of the infection. So we started with Iraq, Dominican Republic, Moldova, Ethiopia, Cameroon, Kenya, Nigeria. So all these countries, we managed to sequence a certain amount of circulating viruses, just providing this information and not only the information, but also providing the workflows for this kind of technology to be able to be performed in the country. Together with the sequencing, we also do the isolation. And here is an example, because this is the first wave is shown on the top. And this is a typical virological technique, which is called plaque assays, where you take a cell monolayer, you dilute the virus. A virus infects the cells, and the surrounding cells, they detach. So then you can fix the cells, and you have holes that you can count. So each white plaque represents one virus, and you can have a measure of the infectivity, so how many infectious particles you have. This is not comparable directly to the physical virion, because the physical virion can be defective in terms of infectivity, but is a measure of the number of infectious viruses that are present in the culture. And interestingly enough, so the first wave, this was in March 2020, you see very nice round plaques. Already in the summer of 2020, when everybody thought that the virus had disappeared, in fact, it was still circulating and already has changed in its behavior. So you can see here that even though you have virus, so the virus is present, you don't see the plaques. So sometimes the plaques, the formation of the plaques, the ability to infect a cell and make it cytopathic and detach is a feature of a virus that can change in time depending on the genotype. So the relationship from the genotype to the phenotype is one important aspect of biology because it determines also its pathogenicity somehow. So this is an overview of very basic techniques that we are doing in identifying and surveying the virus that was circulating in different countries. Another program was the development of antiviral drugs. As I mentioned, isolating the virus is very important because you have the virus, and the virus is important because you can test for antivirals. And therefore, we initiated a program to develop antivirals together with partners in different contexts, from university colleagues to companies that wanted to test molecules, other partners in other countries that had different approaches. So we were open to collaboration to test antiviral activity of different drugs. The first test that we use is a typical plaque reduction assay. So as I mentioned before, you can have formation of plaques as a measure of infectivity. If the plaques are reduced by an antiviral, you can measure how much the reduction is in terms of with respect to the concentration of the drug, and therefore have a readout in vitro of the activity of a drug. This doesn't mean that the drug is going to be effective into the clinics, and this is a very important point because, for example, hydroxychloroquine, which is a drug that works perfectly well in vitro, so we use it as a control because it's cheap and you can quickly use it. In fact, in a clinical setting, it has been proposed as an antiviral drug, but in fact, it's not so effective. So you have to be very careful in extrapolating data from in vitro to the clinical setting. Nevertheless, we were able to initiate this assay initially as a plaque assay and then as a more high throughput assay together with Luca. So we developed an antibody to detect the virus. So we had also the reagents that we were prepared in-house. This is a recombinant antibody that we produced and is used to detect the spike protein. And we have an automatic setup for, let's say, medium-high throughput analysis of antiviral activity. And this helped a lot us into testing more and more compounds. Of all the compounds that we tested, some went on into clinical trials, in particularly the AQCH, which is phytopharmaceutical, is a compound derived from plants that was developed in India by a company, Sun Pharma, together with our laboratories in India, in Delhi, for dengue. And we tested for SARS-CoV-2, so it's a kind of repurposing drug because it was developed for a different virus and then showed to be effective also with this for this infection. And this went on to phase 3 clinical trial. But also we identified other drugs that are repurposing drugs. So again, drugs that are already available in the clinics that can be used for SARS-CoV-2, like miglustat, selgosivir. Selgosivir is a preclinical drug. We have nitroxolin, which is an antibiotics, and so on. So we have a certain number of drugs that we also patented and at different levels of activity, let's say of clinical trial, and also a different level of exploration of the mechanism. So for some of them, we were quite advanced in terms of understanding the mechanism. For others, less. And among the tools that can be used for understanding the molecular activity of an antiviral drug, several of the techniques that will be presented during this presentation that are available here will be pretty useful. I'm talking about electron microscopy, but as well as other techniques as synchrotron. It's important sometimes to help us to understand the mechanism of action of different drugs. One important aspect of drug discovery is that these drugs, tendentially, we don't look for drugs that target a specific feature of the virus. So, for example, now the drugs available for SARS-CoV-2 are targeting either the protease or are targeting the polymerase, so enzymes activity of the virus. And this is a classical example of an antiviral drug. But we focus mostly on indirect effects, so drugs that target functions of the cell that are then required for the virus to be infectious. And this is, to us, is an approach that, of course, is less efficient in terms of targeting a specific virus, but opens up to having broad-range drugs. And this, to us, are important because in the verge of an emergency, what you need is broad-spectrum drugs rather than a specific drug. You need time to develop a specific drug. While if you have a battery of drugs that are targeting different classes of viruses, then you can cover and maybe have a first line of defense against an emerging infection. And this, of course, also occurred also for SARS-CoV-2 because at the beginning, if you remember, remdesivir was used a lot in the clinics, and remdesivir is a drug that was not developed for SARS-CoV-2, is a repurposing drug, and therefore was used because it was shown to be effective. So if you have a battery of this kind of drugs that you already know that are effective on different classes of viruses, you have something at hand at a very early stage of an emerging pandemic. An important aspect of our work that I didn't realize at the beginning was a pressing request from industry to provide proof of disinfection and treatment of the virus in the environment. I mean, we are all here in a seminar room, You remember at the time of COVID, this was not allowed, and was not allowed because the circulation of the virus in this room would be dangerous, and everybody would pick it up except the one wearing a mask that is protected. The point is that these environments were not designed to protect from a circulating infection, and we're not talking only about SARS-CoV-2, but also flu and other infectious diseases that are transmitted by air, so that are potentially harmful in this context, can be treated by having tools to disinfect the environment. So the air circulation can be disinfected by UV, for example. The LED can be engineered to have an antiviral wavelength and so on. And all this was fully developed at the time. And so we received requests to test some of these devices. We collaborated with Fincantieri. Fincantieri was heavily hit by the pandemic. Remember, all cruise ships were hit by the pandemic because this was a source of contamination. They asked us to test UV lamps that were then installed in the ships to disinfect the air conditioning. So we did the testing in our BSL3 laboratory. Here a picture from our BSL3 where we tested the lamps. An interesting aspect was a visible light lamp, LED, produced in Italy by NextSense, which is a company based in Rome. And I was very skeptical at the beginning, but this is a LED that illuminates like a normal light, and it has antiviral activity. So you can see here a scheme where we could see that really disinfection of a surface was completely abolished in a short time. So there are solutions that can be implemented to make environments more, let's say, safe for an infection, an airborne infection. And this is also relevant to the platform because, okay, we measure the antiviral potential, so the inhibition of the viral infectivity, but the mechanism of that inhibition is not clear at all, because maybe UV can be effective against the nucleic acid of the virus, and this can be understandable, it can be tested, but about the mechanism of the LED, I still have little understanding of the real mechanism of inhibition of the infection. And for that, some of the tools that are available here could be very important. So I think these are aspects that could be considered for the platform. Of course, the other aspect that we are a lot interested in is diagnostic. So meaning how to detect a viral infection. Here is an example of an assay, which is called LAMP. Is a molecular assay similar to a PCR, is an isothermal amplification. Okay, so this is, I've been working on a point-of-care delivery, point-of-care tests for diagnostic of viruses for a long time. Point-of-care means tests that you can deliver to in the field or at the bedside. So not tests that you do in a centralized laboratory, but tests that you can use in different settings. Of course, we're very interested in tests that can be delivered in Africa in the field, because places like Europe, you have centralized facility for diagnostic in every city, so in every hospital, and therefore you don't need a distribution, a capillary distribution of tests on the territory, while in Africa you may have large portion of the country that are completely void of healthcare facilities, and therefore you need to have tests that can be delivered in the place. So a test to be run in this context has to be very simple, and this is a test that we didn't develop this test. We collaborated with a company doing a biolabs who developed the test to transfer this test in Africa. And this is a simple test. It just turns pink to yellow. It's a colorimetric assay. If you have the reaction, a positive reaction, you have this dye that turns to yellow because of the pH of the reaction, and has been proven to be extremely robust as an assay, and compared to RT-PCR. And these are the data from a large multicenter clinical trial that we performed in 4 countries in Africa, among 1,500 patients, to show that actually this test could be comparable to RT-PCR, a bit less sensitive, but compared to RT-PCR for the detection of SARS-CoV-2. Why I'm saying this in this context? This is important. This is a successful example of a test that can be delivered in this context. It has a number of advantages because it's colorimetric, it's simple to do. You don't need don't need to have RT-PCR, you don't have a thermocycler, you just need a water bath and so on. On the other hand, it has a problem, like all these tests that are colorimetric or the typical antigenic test that we all did with the the lateral flow signal are tests that are difficult to record because they are not automated. So you lose a lot of information because you do the test and then you throw it away. There is no way of recording unless you take a picture or you record it. So somehow these are excellent, but are they're difficult to monitor. I'm saying this because in the past, we collaborated with different companies, different solutions for developing point-of-care assays. This is an example of a work that we did some time ago in the context of a FlaviGov, which is a project that involved different stakeholders. Principally Proxentia, which is a company, a spin-off of the Politecnico di Milano, and they developed this assay, which is called reflective phantom interface. It's based on reflection, and so when you have the binding of an antigen to an antibody, you can see a change in the reflection of this surface, and therefore you can monitor the interaction, and therefore you can have a readout of in this case, a direct readout of the binding of the antibody, and therefore can be used to develop a point-of-care assay. And they actually developed that with these cartridges and the reader that can be applied to a mobile phone. So this kind of assays work very well. They work in the lab, in the context of a lab or a testing in a situation that is not in the field, but become very, very complicated to operate in the field. So it's always an effort to develop a test and to be able to transfer it in the field. So this aspect has been a lot missing in several of the tests that we investigated with different partners, and the only one which was successful was the RT-LAMP with this assay from NEB, which actually proved to be very, very efficient. So the trade-off between being able to be transferred and simple to the field and the ability to be able to be miniaturized and technological to have the record is a complicated trade-off. So sometimes you need to be simple, sometimes you need to be more sophisticated, and so this is also open to more investigation. About molecular biology and the basic aspect of infection, I will not go too much into the detail, just to show a recent work that we did in collaboration with IFOM in Milan, with a colleague, Fabrizio Daddak, who's expert in senescence and double-strand break repair. So we basically discovered that the virus is able to trigger double-strand break repair, so it actually triggers a response in the cell that is similar to a senescent response, and also blocks by different viral proteins aspect of the pathway of repair of the damaged DNA. It's a very complex set of experiments. It took me 2 hours to explain it because it's a lot of work. But basically, the point is that we also investigate basic aspect of infection, and sometimes we need tools that can be that we can find in the platform to help us and investigate different pathways. An example not related to SARS-CoV-2, related to some old work on tick-borne encephalitis, which is an arbovirus used, for example, EM tomography to reconstruct infected cells, and typically tick-borne produces this kind of vesicles that are present in the endoplasmic reticulum where the virus replicate. And similar, I mean, SARS-CoV-2 does the same, although the vesicles are in a different shape and different mechanism, but still the concept, if you wish, is pretty similar. Where these are viruses that have an RNA as a genome, And having the RNA in the cytoplasm of a cell is a danger signal for several responses of the cell, and therefore protecting the replication into a vesicle is a way that the virus exploits to kind of protect from this kind of antiviral signal. So again, ways to investigate different aspects of biology. So I think I will stop here. I just give a very brief overview, and I'm happy to take questions. I think it's important that we do this kind of activity to learn what each of us is doing, also to find ways of collaborating. To me, what is critical is in the collaboration is not only to prove the principle of that alpha technology works, but to address a specific biological question. And that has been always the difficult part. I've been collaborating with some of you in Electra in the past, and we made a lot of effort to develop a technology to detect maybe one feature of a viral infection. And this is fantastic, and it's very important. But this is not enough. We need to address a biological question with the technology. In that case, this becomes the real step forward in our work. I close with some of the partners of our work, and you can see here is a mix of different sources of funding, from private industry to public financing bodies. Really, what we do is at the boundary between translational research and basic research. I don't think we have to distinguish necessarily between the two. We can do both, as long as we keep the value of doing basic research, because everything I think goes around basic research, and that the spin-off of translational research is done with the right collaborators that then take over the work, because we cannot do all the work down the line. We have to stop at a certain point. So I think this is the the good trade-off between two things is what is important to maintain. So thank you very much.
The correct receptors. I mean, how can you have cells which are?
Oh, you're right. So, okay, it's based on experience. Many viruses have a broad range, so don't have necessarily a specific receptor, and therefore are able to infect different cells. And these cells is only based on experience, have been used to grow several viruses because, as I said, they are defective on the antiviral response, basically. So if the virus finds a way in, it replicates. And this has been used for, you can isolate herpes virus, you can isolate SARS-CoV-2, you can isolate rabies, several viruses. You cannot isolate HIV, for example. HIV is completely refractory to this. There's no receptor. So some viruses will never grow there. It's one of the ways you use to isolate a virus. Not the only one, but it's one of them. To me, it's important. Isolation of the virus is important because a lot of virological research is done by detecting the nucleic acid. But this is not enough. I mean, detecting nucleic acid is important, but doesn't give you a real understanding of the infectivity. It's only, one aspect of it.
The E6 have been used everywhere, and are considered the way to understand if the compound or if a drug can have an effect on the virus. In the meanwhile, I think we understood that this is not exactly the very meaningful information that you have to test other things in parallel. Do you have an idea or suggestions of what else should be done ab initio when you have a series of potential compounds or whatever that can have an effect that are beside following the infectivity?
Okay, so the question is about antiviral drugs and how to develop an antiviral program, discovery program, I would say. So I think the first step is set up a screen, high-throughput screen based on infectivity. I think is very critical because, as I said, it's important to inhibit infectivity as a first starting point. And there you can already have a lot of information because you can use different ways of adding the compound to the cells to understand the mechanism of action. So you can add the compound before infection, after infection, so you can have an idea of where the compound is targeting, so what stage of the infection is targeting. and also the level of inhibition, if it's in the micromolar, nanomolar. If you're looking for an antiviral drug that targets a viral enzyme, if you are in the macromolar, forget it. You just can throw it away. It doesn't work. It doesn't help. While if it's a broad range antiviral drug, if you're in the low macromolar, it has potential. So these are two elements that you can already derive. Of course, Vero cells are a starting point. We worked, for example, on inhibitor of cathepsins, and the level of this enzyme this is a cellular enzyme. This is essential for some aspects of viral entry, and its expression in the cells can vary. So in Vero cells, you can have, I don't know, more cathepsin B or less cathepsin L, while in HLH7 cells, you have the opposite, or you have CALO3 cells. So when you are targeting a cellular aspect of the infection, that is critical for the infection, the different cells may have different activity of different pathways. Therefore, having a range of cells to use is important. Vero is a starting point. CALO3 probably for SARS-CoV-2 is relevant because they are a model of the real target in the airway epithelia of the infection. HLH7 cells are also used very often in this kind of analysis. Reviewers tend to ask you always a primary cell, or maybe a primary airway epithelia, or some kind of primary cells, just to show that you can prevent infection also in a more physiological context. Okay, these are the steps in terms of in vitro. Then, of course, you need to do preclinical testing in an animal model. So that's why we also want to use animals in the BSL3.
Thank you for the overview of your activities and interesting points on different study you can perform, both with research partners and companies also. I was interested in knowing what was the time scale you used in testing the antiviral efficacy of UV light, because it was mentioned. Maybe you can go back to the slides so that we can understand.
Yeah, but there's a no, I know, but there's a delay in the. Okay. So here, so
I guess that there is a time scale, so there's 5, 15, 30, 45.
Yeah, the time scale. And here is PFU per ml. The readout here. Can you see the pointer? No, I don't see the pointer. The readout here are PFU, so plaque forming units per ml, infectivity.
Okay.
And this is time scale. So you can have a decrease of the infectivity.
Are those days or hours?
Hours. Sorry, minutes.
Minutes.
These are 5 minutes, 15 minutes, 30 minutes.
Okay,
yeah.
so
not very readable.
Effective, because I mean, if you talk about days, it wouldn't be applicable instead.
So
effective.
Yeah, so just to summarize, the question is about the use of UV light or LEDs for antiviral activity. How we operate with the industry. It was, during the time of COVID, it was a little bit hectic. So we have the request. They didn't know exactly what they wanted. They wanted to develop workflows and protocols for sanification of cruise ships. Cruise ships are enormous, so you can imagine, develop a protocol for that is really challenging. So they had 3 elements. One, they wanted to have treatment of air conditioning, so the air that was circulating in the ship. Very important, because that is the source of contamination. Second, they wanted to have ways of decontaminating surfaces. Okay, that's easier because you can have different ways of decontaminating a surface in the different part of the ships. And then they want to have something that they could use in a populated environment, because UV, of course, you cannot use in an environment where you have people They want to have a system that they could have without having a problem with the people. So for the treatment of the air conditioning, so they had these UV lamps. It was huge, it was this size. So they delivered us one of these lamps that you could test it. So it was, I can imagine, it was complicated to test it. So we put this in a hood in the BSL3, and then we developed different distances on the surfaces to understand how effective it was and the time of loss of infectivity. Because then then they put an array of this UV in the pipes of the air conditioning that are 2 meters diameter. They are enormous. And then the engineer calculated the time a particle could cross along this grid to get inactivated. So by this calculation, we could have an estimate of the ability of this device to decrease the infectivity. Okay. It does kill many things, but you need to test. Also, that is an aspect that I was pretty naive in this aspect because I never worked on this aspect before COVID. If you want to market a device for the contamination, there are some standards that you use to prove that your device is working. But these standards are based on a parvovirus, so a canine parvovirus, or M13 phage. So these are surrogate viruses that are used to prove that a device works. But nobody wants to know if it's effective against a parvovirus. So they want to know if it's effective against SARS-CoV-2. SARS-CoV-2 luckily is very sensitive to inhibition, to be honest. You can kill it very, very easily, but I think there is a lack of the right standards and procedure for doing the inactivation. And one of the things that we would like to do in the future is not to, because here we had to work a lot on surfaces that where we put the virus, because to spray the virus in an environment in the context of an experimental setup is very dangerous. You cannot spray it in the hood or in a room. Or, I don't know. It's difficult to control, and it's difficult to have a procedure. So you need some closed boxes where you can spray the virus and then collect the virus and see if you have an inhibitory. So we did it on surfaces. Our next step would be to develop devices for testing aerosol, and that would be more important. But yes, this would be effective against all pathogens. How much energy you need and how much time you need for inactivation depends on the pathogen. Yeah.
So we are developing plasma cell membrane models in which we mix in different proportion, mimicking the real plasma cell membranes, lipids and protein component. We are going also to introduce transmembrane protein in that. And we can test the pathogenicity of viruses. We work a lot with extracellular vesicle. Now we have a modified version of the population of extracellular vesicle, which have the SARS-CoV-2 spike and test the pathogenicity and so on. We could think of testing this platform also in the presence of antiviral stuff. Are you interested in this type of interaction? This is my first question. The second question is just a curiosity regarding the device that you showed for antigen antibody interaction. I didn't understand how the antigens are
so there is
on the surface.
Okay, so the second question first. This is the Proxentia device, and there is a glass that is coated in a way that is very transparent, and for the reflection so the way is basically absorbed on the glass surface with a protocol that from the CNR of Milan, they had a specific coating of the glass to deposit the spots of the antigen for the reaction. No, I don't. No, there was no coupling, I think. I can. I think it's quite a specific. Yes, I can give you the details because
It's reproducible, this type of.
Yeah, I can give you the details. Yes, that was made on for that reason. Yes. The other question was related instead to the models of plasma membrane. This is very interesting because I put on the slide now because the last of the compounds that you see is this benzothioazoles. These are virucidal compounds that we identified. They are potent antivirals, not only for SARS-CoV-2, but for all membrane viruses. You know that you have viruses that are surrounded by a membrane and viruses that are not. So these are all this is active against viruses that are surrounded by a membrane, so not only SARS-CoV-2, but also others. And actually, the mechanism is related to the disruption of the membrane. Well, by electron microscopy, we could see that the virions appear kind of elemental cheese. They have holes in the membrane. And we also we use a model that basically was done in Milan, a model of membranes, as you were mentioning. I don't know what is the difference between your model and their model, whatever, to study the effect of these drugs on the membrane structure and how this is perturbed by the drug. We are also collaborating. Dennis at the back of the room is collaborating with Barbara Rossi at the Electra for the use of Raman spectroscopy, and also. Right. But with them, we are able to have a signature of a virus, a membrane virus, and using this same drug, we can see disruption of the signature probably related to membranes. Now, we can discuss how. So this is again, as I said before, we have a technology, we have a question that now is try to understand what kind of data and responses we can get from the technology to improve our understanding of the mechanism. Yeah. Okay.
Thank you very much.
Thank you.